Welcome to the next episode of the Microbiology Tube. So today we'll be talking about the Staphylococcus aureus. So Staphylococcus aureus is the pathogenic bacteria which are mainly found in the pus. So whenever a persons are infected by the wound or have the wound infection, the majority of the bacteria isolated is the Staphylococcus aureus. So there is a rise in concern about the Staphylococcus aureus because the Staphylococcus are becoming more and more resistant in these days. So if you see, the methicillin resistance Staphylococcus has had already been emerged, and now there is the emergence of the vancomycin resistance Staphylococcus aureus. So it is difficult to treat to those kinds of the infections caused by those bacteria. So now I am going to define you a simple definitions or simple background of this Staphylococcus aureus. So firstly, if I start about the Staphylococcus aureus, then I will start about the Staphylococcus. So Staphylococcus means Staphylo, as we have already mentioned, the staph is like a grape insect. So if you see here, all these figures in the figure, if you see, all these are cocci in shape and these are cluster in shape. So such like the bacteria, such like the crabs, these are cluster in shape. So if you see the coccus, if there is written the coccus, majority of the cocci or the coccus bacteria are the gram positive, except there are some of the cocci bacteria like the Neisseria and the Brahmanella. So these are the gram negative, but the bacteria like the Staphylococcus, Streptococcus, Micrococcus, Enterococcus, so these bacteria have coccus at the last, and these bacteria are gram positive. So you can remember by in that way. So if you see, Staphylococcus aureus are the spherical in shape. These are the gram positive bacteria, and the aureus means that is the yellow in color. So yellow in color in term of the colonies. So Staphylococcus aureus will produce the yellow colonies in the nutrient agar. So it was first observed by the von Reckling-Hansen in 1871. So he observed that bacteria, Staphylococcus bacteria in a pus. So Staphylococcus aureus, it is a pathogenic bacteria. Staphylococcus saprophyticus is the pathogenic bacteria, but Staphylococcus epidermidis, Staphylococcus, these are not the pathogenic bacteria. So if you see here, the Staphylococcus aureus produce the yellow colonies, and it is a pathogenic bacteria. So whereas the Staphylococcus alvus, that produces the white colonies, and these are not the pathogenic one. So I doesn't mean to say that yellow colonies are pathogenic whereas the white colonies are non-pathogenic. It doesn't mean that. So I'm just telling how the scientists of that is differentiated the Staphylococcus aureus and the Staphylococcus alvus. So next is the Staphylococcus are the spherical in shape. They are 1 micrometer in diameter. They are gram positive bacteria. They are non-motile and non-sporing. So it means they do not have the flagella and they do not produce the spores. Similarly, the Staphylococcus aureus has one of the distinguishing characteristics, that is the catalase positive and coagulase positive. Staphylococcus bacteria, all the Staphylococcus aureus are catalase positive. All the Staphylococcus are catalase positive. Streptococcus are catalase negative. Micrococcus are again catalase positive. So if you see if you see any of the gram positive cocci, so whenever you observe the gram positive cocci through the microscope, you have to do the catalase test. If it is the catalase test, then it will be either the Staphylococcus or the Micrococcus. So in order to separate whether the bacteria is Micrococcus or the Staphylococcus, you have to do the oxidative and fermentative test, that is called the OF test. So if you do the oxidative and fermentative test, then it will be clear whether the bacteria is Staphylococcus or the Micrococcus, because the Staphylococcus bacteria, these are the fermentative bacteria, whereas the Micrococcus are the oxidative bacteria. So from these you can differentiate. So after the differentiation, the next thing we have to do is to separate the species. So as I have already mentioned, the Staphylococcus aureus is one of the most predominant bacteria which is pathogenic. So in the clinical laboratory we have to separate the Staphylococcus aureus from other species. So one of the distinguishing features is the coagulase positive. So what does we do in the coagulase? In the coagulase test we put the human serum, and in the human serum we put the colonies of the bacteria. So what will the bacteria do? So the bacteria will convert the fibrin to clot. So after it has been clot, so then there will be the visual clumps will be seen in this way. So the bacteria is said to be the catalase positive one, the coagulase positive one. So if the bacteria is gram positive cocci with the clustering shape, and it is the catalase positive and it is the coagulase positive, it is confirmed to be the Staphylococcus aureus. So the question is, there are others, biochemical test tube by which we can differentiate the Staphylococcus aureus. One is the mannitol fermentations. So mannitol fermentations means that it will ferment the mannitol into the acid. So it will ferment the mannitol into the acid. So many of you have known about one of the medium that is called the MSA medium, that stands for the mannitol salt agar. So that stands for the mannitol salt agar. So what happens is that the mannitol will convert into the acid. So what happens, the mannitol will be converted into the acid, and after the acid, so after there is the acid production, the phenol red indicator will change its color from the pink or red pink to the yellow, golden yellow in color. So the mechanism behind the mannitol salt agar is that there is the mannitol that mannitol will ferment the mannitol to form the acid, and if there is rise in the acidity, then what happens is that the phenol red is the indicator, so it will change its color from the pink red to the yellow in color. So there is also next term that is called the S, which is called the salt. So mannitol salt means there is the presence of the 7.5 salt per concentration. 7.5 NaCl is present. So if you see, there is the presence of the 7.5% NaCl, and I have written here that the Staphylococcus aureus are the halotolerant bacteria. It means it can tolerate the salt. So together, if you see the mannitol salt agar, so Staphylococcus aureus are responsible for fermentations of the mannitol, so it will produce the acid and it will change the color from the red to the yellow. So in that way we can see whether the bacteria will ferment the mannitol or not. The next is there is the high concentration of the salt, and Staphylococcus aureus are the halotolerant bacteria, so it will tolerate the 7.5% of the NaCl. Some of the strains of the Staphylococcus aureus are also there which can tolerate up to 10% of the NaCl. So now if you see here, so it will also reduce the nitrate into the nitrite. So it can also reduce the nitrate into the nitrite. Staphylococcus are the methyl red positive, bogus Poskar positive, and it also produces the enzymes called as the phosphatase enzymes. So next is the Staphylococcus will reduce the telluride, which will produce the black colonies. So in the medium there, if we put the potassium telluride, so that telluride will be reduced and the colonies will be seen as the black in color. So next is the cultural characteristics. So here I have taken the two pictures from the internet. So one is the Staphylococcus aureus in the nutrient agar. If you see here, this yellow colonies is the Staphylococcus aureus. So after the 24 hours of incubation, Staphylococcus aureus will produce the large colonies. So these are circular, convex, smooth, shiny, opaque, and easily emulsifiable. So it will easily emulsify. So next is the Staphylococcus aureus will produce the golden yellow colonies in the MSA MSA. So this is the nutrient agar. So this is the nutrient agar, and this is the mannitol salt agar. So you can see here, so this is the Staphylococcus other than that of the Staphylococcus aureus, and this is the Staph aureus. So here you can see the golden yellow colonies of the Staphylococcus aureus here, whereas here you can see, so whereas the here you can see the other Staphylococcus aureus, which doesn't ferment the mannitol. So now at what temperature the bacteria can survive? So Staphylococcus bacteria or Staphylococcus aureus has the wide range of temperature in which it can survive. So it can survive from 10 to the 40 degrees centigrade, whereas the optimum temperature is about 37 degrees centigrade. So it has the pH requirement of about 7.4, and it has 7.6 is the pH requirement. So similarly it is the aerobes as well as the facultative anaerobes. What does that mean? That means that it can survive in the presence of the oxygen as well as it can survive in the absence of the oxygen. So these are the facultative aerobes and the facultative anaerobes. So the next characteristic is that it produces the clear zone of hydrolysis. So here you can see this is the Staphylococcus aureus and this is the blood agar. So here this is the clear zone of hydrolysis. So next character is that the blood agar can produce the clear zone of hydrolysis. In the blood agar, the Staphylococcus aureus can produce the clear zone of hydrolysis. So thank you for watching my video. If you really like my video, please don't forget to press the like button. Please subscribe my channel and please share the video. Thank you.